Good morning, guys. Today we're going to go over the first 4 weeks of embryonic development. There's a lot that happens in those first 4 weeks. So we're going to be talking about pretty much everything that happens after fertilization up until the end of those 4 weeks. So it's going to include things like the forming of the zygote and blastocyst, the process of implantation, the differentiation of the primary germ layers and the extracellular membranes, the process of gastrulation and neurulation. And so by the end of this, you have to know what all of those processes are and all of the different structures that are going to be developing. So we'll start with the process of fertilization. We've already talked about in the previous lessons how the process of fertilization happens in the fallopian tube when the sperm and the egg combine. The egg, or the ovum, is surrounded by two different layers. The first is called the zona pellucida, and it is a jelly-like protective layer that surrounds the egg. And then the second layer is called the corona radiata, which is a whole bunch of leftover follicle cells that are used as a source of nutrients and nourishment for the developing egg. The egg has to be fertilized within 12 to 24 hours after its release, or it won't develop any further. The sperm is going to come along, and the digestive enzymes in the acrosome of the sperm, the little helmet part, are going to slowly digest their way through the zona pellucida that surrounds the egg. Once the sperm makes it through that protective layer and actually into the egg's membrane depolarizes. And once that happens, no other sperm are able to enter the egg. It's how the egg ensures that it's only going to end up with the correct amount of chromosomes, 46 chromosomes. If another sperm came in, it would have too many chromosomes. As soon as the egg is fertilized and you have the combination of the sperm and egg DNA, we now call that structure a zygote. The zygote is going to have 46 chromosomes, 23 from the egg and 23 from the sperm. Okay, so this is just kind of a diagram of what this would look like here. So all of these kind of pointy cells around the outside are the leftover follicle cells that make up the corona radiata. And then beneath that, this kind of pale section is the zona pellucida. So that's the jelly-like protective layer that the acrosome has to digest its way through. And then once you get through that, this kind of darker pink area here is the actual egg itself. That's all the cytoplasm. And then obviously we have the nucleus there. So once the zygote is formed, it is going to continue traveling down the fallopian tubes towards the uterus. While it's doing that, it's going to undergo a process called cleavage. The cleavage process is essentially the same as mitosis, only the cells don't grow in between dividing. So normally in mitosis, you would start off with one cell, it would divide into two, each of those would divide, but as they're dividing, they divide and then grow back to your original size before dividing again. With the cleavage process, you still have, the original zygote, it then divides into two, those will each divide in half again. But the cells aren't growing after they divide. So the cells are getting smaller and smaller every time they divide. That cleavage process will happen until we have something called the morula formed, which is just a stage in which we have 16 cells all kind of lumped together. And that process of moving down the fallopian tube will take 3 to 5 days, depending on how far the egg was from the ovary when it was fertilized. So down at the bottom here, you have here the process of fertilization, which is when the one sperm actually made its way into the egg. At that point, the membrane depolarizes, so none of these sperm will be able to get in. So that is now a zygote. And then that zygote splits in half. That's how we get the 2 cells. Each of those will split in half. That's how we get 4. Each of those 4 split in half. That's how we get 8. Each of those 8 split in half. That's how we get 16. And so that process of getting to the 16-cell morula is the cleavage process. That morula is then going to enter the uterus, and once it gets into the uterus, it's going to fill with some uterine fluid. And at that point, it becomes a blastocyst. You're then going to have two different types of cells develop in the blastocyst. You will have the tropoblast, which goes around the outside, and the inner cell mass, which is in the inside with all of the uterine fluid. So we can see that in this diagram over here. The tropoblast is the bluish part around the outside. The pinkish cells are the inner cell mass, and then all this gray stuff is the uterine fluid. The tropoblast is then going to develop into the extraembryonic membranes, which include the chorion, the amnion allantois, and the yolk sac. So those are all things that will support the embryo, but are not the embryo itself. And then the inner cell mass will develop into the actual embryo. That blastocyst is the structure that then implants into the endometrium, which is, again, that inner layer of the uterus. So once we have that implantation of the blastocyst, the tropoblast, which is that outermost layer, will develop into two different structures, the chorion and the amnion. The chorion cells are going to be the ones that secrete the HCG hormone, human chorionic gonadotropin. That's the one, if you remember from our menstrual cycle lesson, that HCG hormone is the one that stimulates the corpus luteum to continue producing progesterone so that the uterine lining stays healthy and intact. The corpus luteum, again, is very important. If it weren't there, the flow phase would start, and that would mean that not only does the endometrium get shed, but the blastocyst and developing embryo would as well. After the first trimester, the corpus luteum will then be able to start degenerating because the placenta will take over some of the progesterone and estrogen production. Okay, so in week 2, we start having more development of some of those extracellular membranes. There are 3 extraembryonic membranes, like I said, the chorion, the amnion, and the yolk sac, which are there to support and protect the embryo, but they are not actually part of the embryo. They will allow the embryo to get nutrients, to get rid of waste, to breathe, all of that kind of stuff. They are also going to be expelled from the uterus during birth, but again, not actually a part of the embryo. So the chorion is one of the first ones to develop. Again, in week 1, we start having the development of those chorion cells. By week 2, the chorion starts to create these folds, which we call chorionic villi. And those little folds will start stretching deeper into the uterine lining. As those chorionic villi continue to develop, they will eventually become the placenta. But when the placenta is immature, we just call it the chorionic villi. We also have the amnion forming. And so essentially what that is a sac or a layer that is going to surround the developing embryo. It's going to be filled with fluid, which is the amniotic fluid, and it's there to help insulate the developing embryo, protect it from infection, protect it from dehydration, protect it from any major temperature changes, that kind of stuff. And then the last one to develop, this one actually doesn't even start developing until week 2, is the yolk sac. And it'll start developing just below the embryo, and it is where red blood cells will start to form. Later on, it will also develop into a kind of very early sort of digestive tract. So that whole process is essentially how the 3 extraembryonic membranes form. Those are the 3 extraembryonic membranes. You also, in week 2, have a process called morphogenesis that starts happening, which is essentially the very beginning of distinct structures forming in the embryo. So morphogenesis involves 2 processes. The first is just that the embryo is going to start growing, and the second is differentiation, which means that instead of all of the cells being identical to each other in that blastocyst, you're going to have different types of cells start developing. So you might have some like heart cells start developing, or some red blood cells, for instance. So you're going to have differentiation of different cells with different functions. The very first step of this morphogenesis process is a step called gastrulation. And gastrulation is just how the 3 primary germ layers are formed. So in the process of gastrulation, the inner cell mass, the part of the blastocyst that's on the inside and is going to become the actual embryo, that inner cell mass is going to flatten out into what's called an embryonic disc. And that disc is going to be made up of 2 layers. The outer layer is the ectoderm, the inner layer is the endoderm. And then there is a short little stalk that helps support that embryonic disc that will later develop into the umbilical cord. And then between the endoderm and ectoderm, we get a 3rd layer that's going to form, that is the mesoderm, or the middle layer. Once you have that 3 different layered structure, we now call it a gastrula. So this is what it would look like down here. So this little section in the middle here is the inner cell mass. It has pancaked out into that disc shape. And then it is going to develop into the ectoderm, which will go around the outside, the endoderm, which will be in the middle, or inside, I guess. Eventually, once the mesoderm forms, it becomes part of the middle. You can see that up here as well. So this purple and yellow line here in the middle is the embryonic disc, that flattened out inner cell mass. It is going to develop into the 3 different layers that we can see here. We've got the ectoderm, which is the purple layer, the mesoderm, which is a little pink layer in the middle, and then the endoderm on the bottom. Okay, so these 3 little lines here make up the gastrula, those 3 different primary germ layers. Just another diagram of how this would work. So we have the morula. So this is our 16 cell structure just before it enters the uterus. Then it's going to enter the uterus, fill with some uterine fluid, have our inner cell mass up here, and around the outside is the trophoblast. Again, that trophoblast that's around the outside is going to differentiate into our extraembryonic membranes, which includes the amnion, includes the yolk sac, and also includes the chorion. And then this little middle section here is what all of these inner cell mass cells turned into, is this embryonic disc. The blue layer is going to be the ectoderm right here, just this straight line of blue, not the whole way around, just this straight line of blue here is the ectoderm. The yellow line here is the endoderm, and in the middle where this red is the mesoderm, and those are the 3 primary germ layers. You do need to know what each of the primary germ layers will eventually differentiate into. So the ectoderm, which is again the outermost layer, is going to turn into things that are on the outside of your body, like your skin, hair, fingernails, sweat glands, eyes, ears, teeth, as well as most of your nervous system is all going to come from the ectoderm. Mesoderm, that middle layer, is going to become muscles. It's going to become your blood and your blood vessels, your excretory system, your reproductive system, your bones. All of that is part of the mesoderm. And then the endoderm, the innermost layer, is where the bladder comes from. So that is the only part of the excretory system that is in the endoderm, not the mesoderm. Your digestive system, your respiratory system, and a good chunk of your endocrine system is all going to be the endoderm. This structure here is just showing a very early glimpse at our primary germ layers and our extraembryonic membranes. So here, this yellow part is the yolk sac. The kind of teal greenish area is the amniotic fluid, and the just outside of that is the amnion itself. The chorion is this kind of folded part that is growing deeper into the uterine wall, which is this light pink area over here. So all of that is the chorionic villi. The allantois is what is eventually going to turn into the umbilical cord. So yeah, so those are the three extraembryonic membranes. The yolk sac there, that's where the red blood cells are going to form. The amnion, which surrounds the amniotic fluid, which is going to provide insulation and protection and stuff like that. And the chorion, which is eventually going to turn into the placenta and is releasing HCG right now. And then our three primary germ layers, the endoderm, mesoderm, and ectoderm. We can see this blue area here is the ectoderm, that's the outside. Then we have the mesoderm, which is the red section, and just the bottom of this yellow section, just right in here, is the endoderm. Again, not including this little pointy part, which is going to turn into the umbilical cord, and not including this big ball-shaped part, which is the yolk sac. Okay, so that all happens in the first 2 weeks. In week 3, we then have the process of neurulation start, which is essentially how the central nervous system starts developing. So you'll have a neural tube that is going to form, a neural plate that is going to form, and all of that is eventually going to turn into just the central nervous system. So this is what that would look like. So we have the starting of the neural plate. So the neural plate is just, again, the very first step of the central nervous system forming. It is going to start to fold until it forms a tube. So for instance, it would just be something that starts off flat as a neural plate, and it starts folding and folding and folding until you get a tube in the middle. Okay, that becomes the neural tube. The part that doesn't form this circle, so any part that is left out of the circle, so like this part over here, is going to form like bones and stuff like that. It could form part of like your cranium or part of your vertebrae that surrounds your central nervous system. And then week 4, you start having those blood cells from the yolk sac maturing and filling your blood vessels. Again, the blood cells are coming from the yolk sac. The blood vessels themselves will be made from the mesoderm. The lungs and kidneys also start to develop at week 4. You start seeing just small little like nubs or buds where the arms and legs are going to be forming, along with little tiny buds for where fingers and toes will be forming. You'll start getting a distinct head shape and start seeing very early signs of like eyes, nose, ears forming on that sphere of a head. The brain starts to develop and become visible, again, from that process of neurulation in week 3. And then by the end of this first month, by the end of week 4, the embryo is about a centimeter long. It is 500 times larger than it started off being as a zygote. This picture here is really important. Expect to see a picture similar to this on any types of assessment that we have for this unit. So this structure in the middle here is the embryo itself, which was created from those 3 primary germ layers, endoderm, mesoderm, and ectoderm. This would be the head up here. This little structure here is the eye that is starting to develop. At this point, it does still have gill slits, which will go away, but you can still kind of see them here. This would be the little bud that would eventually turn into the arm. We have a little bud here that would eventually turn into a leg. There's still a little bit of a tail at this point. Again, that's also going to go away. Here we have the umbilical cord. This yellow part here is the yolk sac. Again, that's going to be where the red blood cells are developing. All of these folds are the chorionic villi, which again are producing our human chorionic gonadotropin and will eventually turn into our placenta. And then we have the amniotic fluid, which is this pinkish part, which is surrounded by the amnion or the amniotic sac. Okay, so yeah, there's not a ton of growth that happens in this first 4 weeks as far as like actual size goes. A lot of those changes are going to happen in the following 4 weeks. But you do need to know, again, that process of cleavage, fertilization, implantation, cell differentiation, the structures of the zygote, the morula, the blastocyst that actually implants. You need to know how the trophoblast turns into the 3 extra embryonic membranes, chorionic villi, yolk sac, and amnion, and how the 3 primary germ layers, the endoderm, mesoderm, and ectoderm, are formed during the gastrulation process. And you should also know what neurulation is, which is, again, the formation of the central nervous system. Okay, so this is kind of a summary of some of the different things that are happening in those first 4 weeks. So we start off with the sperm and the egg coming together, and that is the process of fertilization, which is going to form our zygote. That zygote is then going to undergo the process called cleavage in order to form the morula. The morula then enters the uterus, where it fills with uterine fluid to become the blastocyst. The blastocyst is made up of 2 main parts, the trophoblast and the inner cell mass. The trophoblast is going to differentiate into the 3 extra embryonic membranes. Those 3 extra embryonic membranes are the chorion, the amnion allantois, and the yolk sac. The chorion is going to be responsible for secreting human chorionic gonadotropin, which stimulates the corpus luteum in the ovaries to produce progesterone, which is what maintains the endometrium and makes sure that the uterine lining and the embryo are not shed. The chorion also develops chorionic villi, which is all of those folds, which will eventually turn into the placenta. The amnion is going to develop into the amniotic sac, which surrounds the amniotic fluid, which is around the embryo, and the allantois is going to turn into the umbilical cord. And then the yolk sac is going to be producing the red blood cells. And then the inner cell mass is going to differentiate into the 3 primary germ layers, which is what is going to make up the actual embryo. Those 3 layers are the ectoderm, mesoderm, and endoderm. The ectoderm is going to be responsible for skin, hair, ear, eye, teeth, and nervous system. The mesoderm is responsible for muscles, blood vessels, bone, kidney, and the reproductive system. And the endoderm is responsible for the digestive system, respiratory system, bladder, endocrine glands, and liver.